Hello friends, welcome to the channel. So today we are going to discuss about pharmacognosy phytochemistry 2 unit 2, in that Irrioids, other terpenoids and naphthalene. So first, that is Irrioids. It is actually chemically it is monoterpenoid. In general form it is cyclopentanopyrrole, which is found in plant and in some animal. It is biosynthetically obtained from it oxogeraniol, which is found in plant in the form of glycoside, and mostly they are bound to the glucose molecule. And chemical structures such as iridomyromycin, has a defensive mechanism, so it acts against herbivores or against the infection by the microorganism. The next group, that is naphthalene. So it is the class of organic compound structurally related to naphthalene ring. So this naphthalene ring having two isomers. So first one is 1,2-naphthaquinone, second one is 1,4-naphthaquinone, and it does not occur in the form of glycoside in higher plants. So it represents a group of plant secondary metabolite which having cytotoxic property on their ability to generate reactive oxygen species, that is ROS, and it is interfere with the cell respiration. So most important naphthaquinones containing plant belongs to the group of phylogenetically heterogeneous plant families. Next, we will see about the examples. So first example, that is Gentian. So synonym for this drug, Gentian root, Gentiana radix, Gentiani. Biological source, it is dried, partially fermented rhizome and root of yellow gentian, that is Gentinia lutea, family Gentianaceae. So next is collection and preparation. These are the large fleshy roots, erect rhizome. So it is collected from 2 to 5 years old plant, which is by the tugging method, and specially collected in autumn season by cutting longitudinal into slices. So pieces of roots and rhizome, firstly they are white in color, no odor, and during slow drying, on keeping them in heaps and fermenting by slow heating, they get dark or yellow colored and develop the characteristic odor. Next, macroscopy. So color of this rhizome is yellowish brown, which has a transfer annulation on the surface, show conical bud at the top. These roots are narrower but continuous with rhizome. Then it is longitudinally wrinkled and has a circular scars of rootlets. It have a peculiar odor. It gives sweet taste followed by an intensely bitter taste, which having short fractures on the surface. Next is the microscopy. So first is the microscopy of root. So first layer is cork, after that cortex, oil globules, patches of starch, then it containing calcium oxalate, which is acicular raphides, medullary rays, which is multicellular in nature, after that cambium, then xylem vessels, wood parent. So xylem vessels are mostly lignified in nature. Starch grains gives the blue color with iodine. Next microscopy, that is rhizome. It first layer in rhizome, that is periderm. After that secondary phloem, cambium development, and xylem. Then next is chemical constituent. So this drug contains bitter glycoside, mainly known as gentiopicrin, which is also called gentiopicroside. It is crystalline water soluble compound which having bitter value 12,000, and during the fermentation and drying it breaks down into two molecules, that is gentiogenin and glucose. Then chemical test. So for the gentiopicroside, it shows the light blue fluorescence under the UV. Next, bitter glycoside, that is amarogenetin, which is considered as a bittermost substance which gives bitter taste in even 5.8 lakh time dilution, and most constituents are bitter. The taste mainly due to the amarogenetin, and glycoside contain monoterpene Irrioid also, and the color of this drug is due to the flavonoid alkaloid. So flavonoid alkaloid in this drug, that is gentianin, genticin, which gives yellow color. So this is about the structure of gentiopicroside, gentian. Then uses. So it is used as a bitter tonic to stimulate the gastric secretion. Hence it improving the appetite. So that's why it is used as a bitter tonic. Okay, this is about the gentian. Thank you.